Recording in progress.
All right, I believe everybody has entered the Zoom link for today. To our dear chair and speakers, and to everyone who has joined us from around the world, good morning, good afternoon, and good evening, wherever you may be. And welcome to our Thieme WebCheminar celebrating the impactful career and scientific legacy of Professor Erick Carreira. We'll aspire to honor him today through outstanding talks by leading researchers in the field, chaired by our synthesis editor-in-chief, Professor Mark Lautens, who I will be introducing properly in a little bit. Before we start, I'd like to introduce myself. I'm Jeanne Andres, and I'm the executive publisher of the journals Synthesis, Synlet, Synfax, and Synopen. I joined Thieme fairly recently, just over 9 months ago, but I have more than 12 years experience in science publishing. I also have a bit of a background as an academic in a university prior to obtaining my PhD in chemical engineering at the University of Cambridge. I'm absolutely delighted to be here with you today for this webinar, and I'm looking forward to the great talks we will be hearing from our speakers and the discussions that we will have during the Q&A. To start with, some housekeeping items. Please do note that this webinar is being recorded and that for continuity and simplicity, the participants' microphones will remain muted throughout. We do invite and very much encourage you, however, to please use the Q&A window to ask questions, as there will be time for these following each presentation, during which our speakers will respond to as many questions as possible from the chat. We also invite you to join the conversation online via LinkedIn by perhaps sharing your thoughts and takeaways from the webinar. And please also subscribe to our webinar series so that you are kept updated on all the great webinars that we are offering for the rest of the year and beyond. And please connect with us by subscribing to our newsletter as well. My colleague, Dr. Ilya Sharanov, will be sharing the links to these in the chat shortly. Before we begin, please allow me to introduce you to Thieme. Thieme is a company based in Germany, but with a significant presence worldwide, including offices in New York, Delhi, Rio de Janeiro, Beijing, and London, and over 130 years of experience in publishing science and medical information. You may already know about some of Thieme's journals, including Synthesis, Synlet, Synfax, Synopen, and Sustainability and Circularity Now. We also have a suite of biomedical chemistry journals, including Drug Research, Planta Medica, and Pharmacopsychiatry. Other Thieme chemistry products that you may already know about are Science of Synthesis and Pharmaceutical Substances. So if you'll allow me, I'll spend just a few moments now to speak briefly about some of our journals. Our journal Synthesis has been the home for trusted advances in synthetic chemistry for over 50 years, publishing trusted and dependable research results, reviews, and practical procedures. Publishing 24 issues a year, it covers all fields of organic chemistry involving synthesis, including catalysis, organometallic, medicinal, biological, and photochemistry and related disciplines. Our highly respected editorial board members shown here on this slide, led by Professor Mark Lautens, they take care of the peer review process for the journal. Synlet, for early stage ideas and bold new directions, Synlet is our international letters journal, publishing 20 issues a year. If you're looking to get your first findings or new directions out fast, Synlet is designed for preliminary findings, short reviews, and emerging concepts in organic synthesis. And similar to synthesis, peer review is handled by our Synlet editorial board member, shown here, led by Professor Deb Maiti. We publish Synpax articles as well in Synlet, which amplifies and encourages the voices of early career researchers, and the journal is integrated with ChemArchive as well. SynOpen is our fully gold open access journal featuring key reads in synthetic chemistry, led by its editor-in-chief, Professor Thierry Olivier. Aside from research articles, SynOpen publishes graphical reviews, a unique and novel article type that offers visual insights into important topics in the field. With a very high and rapidly increasing readership, which is up 84% over last year, the journal is fast becoming a journal of choice for researchers in synthetic chemistry who want to maximize the reach and impact of their research. Sustainability and Circularity Now, or SC Now, as we call it, tackles the most serious challenges pertaining to sustainability and helping to shape an ambitious and interdisciplinary agenda to resolve the global crisis that we face. So we're proud to say that SC Now has become the first journal signatory of the Stockholm Declaration on Chemistry for the Future, joining a global community in this shared commitment to a better future. We will be having a virtual summit in November as well for circularity, for SC Now, so Sustainability and Circularity Now. So stay tuned. And just to mention before I end that aside from all the benefits of publishing in these journals, each paper published qualifies for a chance to be considered for the annual Best Paper Awards, which are selected by the respective boards of the journals, with the winners recognized the following year, and they receive prize money and a certificate. So on that note, I'd like to thank you for your attention so far, and we will now proceed. It is my special and distinct honor to introduce our esteemed chair for this Cheminar, Professor Mark Lautens. Mark Lautens is the J.B. Jones Distinguished Professor and holds the AstraZeneca Endowed Professorship in Organic Chemistry. Professor Lautens joined the faculty at the University of Toronto in 1987 and has held the title of University Professor since 2012. He has received numerous academic awards and honors for his research, and in 2017 was awarded the J.J. Barry Smith Doctoral Supervision Award, which recognizes outstanding performance in the multiple roles associated with doctoral supervision. Professor Lautens is a Fellow of the Royal Society of Canada and an Officer of the Order of Canada. Professor Lautens holds a BSc from the University of Guelph, a PhD from the University of Wisconsin-Madison, and did postdoctoral work at Harvard. He received an honorary Doctor of Science from the University of Guelph in 2016. Professor Lautens is our editor-in-chief for the journal Synthesis and has been serving on the journal's editorial board for 26 years, for which we are truly grateful. Professor Lautens, Mark, on behalf of your Thieme family, we thank you for sharing our Cheminar today. A very warm welcome, and I now hand things over to you.
Thank you very much, Jeanne. So what I'd like to do this morning is to give you a brief presentation. And as you will see, there's both a very small scientific content and a bit more of a personal content. So one thing I'll say is that there is a unique connection with all three of our speakers today. And I want to begin by thanking each of them for agreeing to speak. They're all people who Erick identified and requested to speak at this event. And they immediately said yes when I wrote to ask them to do so. As I looked through their bios today, I also realized there is a common thread, I suppose, with all three of them, as well as Erick and me, and that is that Harvard figures prominently in one way or another in our scientific and academic journeys. So that's why I begin with this slide, just in a place very far, far away from many of you, since I know that we have people from all parts of the globe. There was a place called Harvard, and light years ago, which was for me 1985, I joined the Evans Group as a postdoctoral fellow. I'm going to get this thing to please advance. There we go. When I joined the group, Dave Evans put to us a challenge. The challenge was how to make the natural product, which had been recently isolated, known as bryostatin. Now, Erick, as you will know, is both an outstanding chemist, but also has a good sense of humor. And this molecule wasn't that easy to make, and it developed the name internally bryostatic because we had some trouble to get to the end. So we began in 1986, as I said, soon after I arrived, and eventually we succeeded. Now, we is a complicated thing in the academic world. So a decade after I departed in 1987, and you can see in 1998, a paper appeared. That was the asymmetric synthesis of bryostatin. And really, it's an interesting collection of people. The person who finished the project was Percy Carter, but really one of the standard bearers of the entire synthesis was Erick, but it also included André Charette, now a professor in Canada, and Joël Pounet, who was a professor in France and now is in Scotland. So this paper appeared. I was very pleased to see my name on it and very pleased. And of course, my students thought it was quite funny that there were all these academics and so on this publication. So of course, Erick went on to do many great things in total synthesis, in catalysis, and really one of his most lasting legacies is in the training of the next generation of academics. And at least one of our speakers today fits into this category. Of course, Erick left Harvard, completed his PhD, went to Caltech as a postdoc, and then spent about a decade at Caltech, followed by 25 years at the ETH Zurich, where he remains today and where he has an active and vibrant research program. So Erick joined Thieme actually before me. He was the editor for the Americas of Synthesis. And when he moved from Caltech to ETH, Thieme came looking for an editor for the Americas, and I was fortunate to be contacted. In fact, Erick was visiting our department to give a lecture back in 1999, and that was when the then managing editor of Thieme came to my office. So I met with Erick. They offered me the job as an editor. And then a few years ago, as was mentioned, I became editor-in-chief. But of course, Erick's done far more than that. When he moved to the ETH, he became very involved with special issues and special topics for synthesis. And in the last decade or so, he's also been a contributor to Synfax. He and his graduate students highlight some of the best of total synthesis. And he's been on the board for Science of Synthesis and been recruiting volume editors and so on. So really today, this is not a farewell, a total farewell. This is really a farewell on his many roles in Thieme, but in fact, he's continuing on as far as I know with Synfax. So we're really here to thank him more than to say goodbye. Now, it seemed to me that it was worth showing a few pictures since I've known Erick since 1985. I've selected a few pictures starting over the decades. In 2009, I went for the very first time to China. I took my son, who you see is the young man in the middle, and Erick was speaking at a Lilly symposium. We had an amazing dinner, eating some very unusual things, and both my son and Erick and I more or less tried everything. This is a picture from one of our editorial board meetings. Now, it's an interesting collection of characters, and perhaps the person with the biggest smile is someone whose face is not familiar to you. That's the gentleman holding the bottle of wine at the back. This is actually the owner of Thieme, and one of the things that's terrific about the publishing house is that the owner is deeply engaged with the publications, with the journals, and is also a terrific human being, and as you can see, has a bit of fun in him, and happens to like wine. This night was the night we actually contacted Franziska Schonebeck to join the journal as an editor, which she did. Unfortunately, later on she left, but this was a happy moment because we were celebrating that she had agreed to that. Sometime later in 2019, one of our board meetings just before the pandemic was in Prague, and there you can see Erick along with a few other of our distinguished editors. At the time, we had a very nice time. I can't remember what I said to Erick, but I'm sure it was extremely funny. Sometime later, we also went to Russia. My colleague Andrei Yudin was our host. we participated in the 100th anniversary of the Markovnikov Symposium, and we had a very long walk with students from the Institute in Moscow, and really had an amazing experience. More recently, of course, Dave Evans, our mutual supervisor, passed away, and we had a symposium at the ACS meeting in San Francisco where many of his former students spoke, including Erick, me, and as you can see in the middle between us is Margaret Fall, who's a vice president at Amgen. But at the symposium, my former supervisor, Barry Troche, showed up, and Erick and I had a good chance to chat with Barry and to take a review of both Dave Evans' amazing accomplishments and also Barry's. Now, I must say, things don't always go quite according to plan. This, of course, is a joke, but Erick came to speak last year in the University of Toronto, and I took him up to our farm. We went to a town that has what is claimed to be the smallest jail in Canada. It's only two-person jail, and by some quirk of fate, Erick ended up somehow briefly in jail, but not to worry. He was able to give his lecture exactly as planned. So really we are here today to offer our deepest thanks to Erick for all the things that he's done for Thieme Chemistry. Of course, he's also done so much for the entire field of chemistry. He's a fellow of the National Academy of Sciences in the United States. He's won dozens upon dozens of awards, well-deserved. But for me personally, of students who he's trained, who have gone on to do all kinds of things. Of course, he's done an awful lot for Thieme, but I cannot help but say that he actually did something for one or two other journals as well. And as many of you will know, he's the editor-in-chief of JACS and travels around the world saying the good works of the American Chemical Society. So on that note, I really want to say thank you. That's the end of my presentation. And so we're ready to move on to the really important part of our day, which is to engage our three speakers today. And as I say, all three, by some coincidence, I suppose, have some connection to Harvard. So our first speaker up today is Professor Tobias Ritter. Tobias was appointed assistant professor at the Department of Chemistry and Biochemical Biology at Harvard. He was promoted professor in 2012. Now, not many people leave Harvard, but if you do, and of course, one of the best reasons to leave Harvard is to be a director of the Max Planck Institute. And this institute I have a little bit of connection with because I spent a sabbatical there when my son was born in 1997. That's the Kohlenforschung in Mülheim, a very famous institute that has more than one Nobel laureate associated with it and has a very long history in the field of catalysis. So Tobias has been a professor there since 2015. And in 2011, Tobias co-founded Cyflor Life Science. It's now called Ocuterra Therapeutics, a clinical pharmaceutical company in Cambridge, Massachusetts. So Tobias, the last time I saw Tobias give a lecture, they had music and a walk-on and a video, and it was kind of like as if either you were a baseball player coming up to see if you could hit the grand slam to win the game or some kind of a rock star. And Tobias is a modern day chemical rock star. He's going to talk about late stage functionalization. So Tobias, I give it to you.
Well, thank you so much, Mark, for that interesting introduction. I cannot offer music, but I hope I can offer a few interesting chemistry slides. But before I start doing that, of course, lots of congrats to Erick for all his contributions to chemistry and maybe even more importantly to the community. Of course, it's a wonderful pleasure for me to speak in his honor today here. I want to talk about late-stage functionalizations and the reasons why we believe those are important. And so one of the first aspects how we became interested in this field was by trying to make PET tracers, so tracers for positron emission tomography, which is a non-invasive imaging modality that we use to image biological processes in vivo. It's a great technique that's used in all hospitals worldwide, but there are bottlenecks for making these molecules that can function as PET tracers, and they have to do with chemistry. One reason is that we need to have a lot of functional group tolerance and chemoselectivity for making carbon-fluorine bonds, because F18 is one of the nuclei that's used in PET. And so here I'm just showing you one example of the work that we have been engaged with, and this is showing you one reaction where we can make these F18 labeled molecules. Now, you have to do it at a late stage because F18 has a half-life of less than 2 hours. So basically, the entire molecule already needs to be assembled before you make the carbon-fluorine bond. Otherwise, the F18 disintegrates and you cannot use it for actual imaging. So this is one reason we think, or we know, late-stage functionalization is important. Now, this is a nice first example, but it really didn't solve the problem, most prominently because it's too complex. It's too complex in chemistry to be used as a standard in a hospital setting. High-valent transition metal compounds, complex chemistry. So we developed simpler chemistry, showing here everybody can handle a phenol. This is chemistry that, in fact, was even used at some other groups and hospitals than ours, and it's been validated for human use for F18 incorporation. And that's okay if you have a phenol available. I think this qualifies still as a late-stage functionalization reaction. But most molecules that you would like, or that we would like to functionalize, like this phenol, right? So when you have the phenol and you can do this deoxyfluorination, even more complex structures, such as peptides or so, you can incorporate this F18. But if you do not have the phenol, then you're out of luck, and you cannot really use this practical method. And so therefore, we developed CH fluorination reactions directly, which is wonderful because you can start from a CH compound directly, so you do not need any pre-functionalization. But the price you pay for that is that you get constitutional isomers, right, as you would expect for an electrophilic aromatic substitution reaction. So you get mixtures of products, and those have to be separated, which in general is not ideal, but especially in the case of aerofluorides, is challenging because these have very, very similar physicochemical properties. So purification of different constitutional isomers of fluorides is time-consuming and difficult, and certainly not acceptable if you want to make an F18 labeled molecule and then have to spend a long time separating these molecules. So what we then became interested in is developing more selective CH functionalization reactions of arenes to solve this problem of generating multiple constitutional isomers. And more or less by accident or by discovering a reaction for the wrong reasons, we became eventually aware of this heterocycle thioentrine and developed reaction chemistry that can incorporate thioentrine into functionally complex small molecules. So here I'm showing you 6 examples of that. And in all of these examples, we were not able to identify any other constitutional isomer of these arenes. So it's an incredibly selective transformation, and there's good reasons for that we try to understand, which have to do with the steric and electronic properties of the active thioentrine reagent that does these chemistries. Now, putting a thioentrine on a molecule by itself normally is not very interesting, but the utility comes out and when you see what you can do with the thioentrine substituent. And here I'm showing you 8 different areas. We have mostly been involved in developing the first 2, so the transition metal catalysis and photoredox catalysis. there have been, over the past 5 years, many other groups around the world who have pushed this thioentrine substituent in utility in a variety of other fields, such as in mechanochemistry, biocatalysis, charge transfer chemistry, because it's charged. So there's been a lot of use of this heterocycle. And what I want to do today is I want to show you 3 examples of how thioentrine differs from all the other pseudohalides. And I want to explain a little bit why it differs and how it differs and why that is important. So here I'm showing you a simple reduction of an aryl bromide by a single electron, which eventually generates this aryl radical. And the aryl radical is synthetically useful because we can use it in high valent transition metal catalysis. If we can generate a copper-3 aryl X, then we can make almost any aryl X bond, and that is synthetically useful, hopefully including aryl fluorides. And that's, of course, well-established chemistry that has been done many, many decades ago to generate aryl radicals from aryl bromides. But it's relatively difficult to reduce an aryl bromide with subsequent aryl bromide cleavage. Now, I'm showing you 2 examples here, an electron-poor aryl bromide and an electron-rich aryl thioentrinium salt. And I picked those just because they have the same reduction potential. That makes intuitive sense because it should be much easier to reduce a cation as the aryl thioentriniums are, because it has a positive charge. So that's not surprising. But what is surprising, or was at least surprising to us, that the rate of mesolytic cleavage, so the breaking of the carbon-bromine bond and the breaking of the carbon-sulfur bond, is 10 billion times faster for the aryl thioentrinium salts. And that has to do with the specific electronic properties of the aryl thioentriniums. It also is the big difference to all the other halides. For example, here I'm showing you a graph where I plot the different reduction potentials over the rate of the mesolytic cleavage, so of the step that generates the aryl radical, which is the important step, right? And so if you have 2 strong reduction potentials, there are such catalysts that can do that, but if the reduction potential is just too reducing, you cannot do high valent reaction chemistry. So everything on the left side of this graph that I'm showing you here is out of the realm of what we can do chemically. At the same time, if the reaction rate is not fast enough for the mesolytic cleavage, then what we have is back electron transfer, and we go back to our starting material because the rate of mesolytic cleavage is just too slow. So that means that the bottom half of this chart here is also useless, and that renders all the aryl bromides and all the aryl chlorides useless for this chemistry, yet puts all the aryl thioentrinium salts, irrespective of substitution pattern, in this quadrant here on the top right, that it can be useful for chemistry. And the fact that they are all in there irrespective of the substitution pattern has to do with the fact that the electronic properties of aryl thioentrinium salts are determined by the thioentrinium substituent, by its positive charge, and not by the other substituents on the array. So then what we can do is we can just combine that with high valent transition metal catalysis. And I'm just showing you one example how we're doing that. Here we can cleave this bond very quickly because the carbon-sulfur bond has a contribution of the SOMO, so of the orbital that is relevant for this breaking, that's not accessible to the carbon-bromine bond. And that is the reason that it breaks 10 billion times faster than the carbon-bromine bond, right? So bromine is just a single atom, whereas the thioentrine is a large molecule or large substituent with that contribution, so that we can access this lower energy, and therefore we can access this mesolytic cleavage at a much, much faster rate. So if you combine then that with high-valent transition metal chemistry, in this case copper, so you do a thiontrination, a regioselective thiontrination, you take a copper source, you take a nucleophile, and then you can make carbon X bonds. And I'm only showing you one example of that here, which is the fluoride, but you can choose many, many other different nucleophiles, and many groups, including ours, have published the work of how to make these different nucleophiles. So that is one fundamental difference to the conventional halides. Another difference is the fact that aryl thiotrinium salts have very different triplet energies, and the aryl thiotrinium radical, or the thiotrinium radical itself, is a persistent radical. So if you look at this aryl thiotrinium salt here, you can cleave the carbon-sulfur bond, and you get two radicals, one of which is persistent, right? You cannot do that with an aryl halide because the chlorine radical or the bromine radical are not persistent. But because the thiotrine radical cation is persistent, it can hang out long enough so that you can add the aryl radical across an olefin, in this case ethylene, for example, and then have the thiotrine radical cation trap the resulting radicals. So you have an addition of two radicals across a double bond. And then the thiotrine is a wonderful leaving group, so you can generate a variety of substitutions from that from the alkyl thiotrinium salt, right? You can use oxygen-based nucleophiles, carbon-based nucleophiles, nitrogen-based nucleophiles, and I'm just showing you here two examples of that. And the reason that the energy transfer is very efficient in this case, because the thiotrine has a very similar orbital overlap as the sensitizer thioxantone in this case. So that's something else that we can do. We can go on the triplet surface, again, something that we cannot do with halides. But even if we go into just the conventional cross-coupling area, which is really the hallmark of aryl halide reactivity, we can elicit reactivity with thiotrines that we cannot with halides. And in this case, it has a different reason. We used it for simple hydrogenation reactions. So this is not a reaction that you would expect to be of importance if you just substitute it with a hydrogen.
Recording in progress.
Step in being able to functionalize selectively large biomolecules and then substituting the selenium, right? Because that is something that people cannot do right now. People can functionalize proteins, people can selectively incorporate substituents, but they then stay, and typically they're very large. What we can do now, similar to the thionetrine chemistry, we can just replace that thionetrine with a single atom, with chlorine, or we can do a Suzuki coupling, or we can do all the other cross-coupling chemistry that we've developed before. And that's something that was not really possible before. If you wanted to have a protein, you just want to add one atom. You have a substitution of one arene and one hydrogen for one chlorine. That's something that wasn't possible before. And while this is not yet a general strategy, it's at least one step towards that area. Another area that becomes accessible now to us is DNA-encoded libraries, where we can now tolerate the DNA with this selenium. We don't have to have the full selenium with the oxazole present. In this case, just the pyridine sufficed. And what we can do here is we can now do CH functionalization directly of DNA-encoded libraries. And we can tolerate the solvent that is required for this, which is aqueous-based, and all the DNA, and then can, of course, use that to functionalize on DNA with cross-coupling chemistry, Menishi reactions, other reactions some of which I've shown you before. Okay, so that is what we've done with aryl thionetrinium salts or derivatives thereof. We can also use other sp2 hybridized compounds, for example, olefins. In this case, we are functionalizing terminal olefins, 1,2 disubstituted olefins, trisubstituted olefins. They all go through these rather intriguing dicationic cyclic intermediates, which are derived basically from a hetero-Diels-Alder reaction of the thionetrine dication, which is the active species, and all of these reactions that I've shown you so far. And once we put the thionetrine there, then we can, of course, do all the other chemistry that I've already shown you, that I'm not going to talk about again here in great detail. I'm just showing you a few examples. Now, my favorite alkenyl thionetrinium salt is this one here. It's vinyl thionetrinium salt, showing you here a few reactions of how it behaves. Basically behaves as a vinyl cation. That's synthetic utility. It's certainly much easier to handle than vinyl bromide is, for example. But one area that is really exciting to us is that it's a soluble and water-soluble salt that can react with cysteines in bioconjugation reactions. And typically, a cysteine bioconjugation is not a very exciting transformation because there's so many that are already available, and they're fast, and they're robust, and they're useful. But vinyl thionetrine can do one thing that's very different than all the other bioconjugation reactions that are out there, and that is that it forms in situ this episulfonium salt, which is more reactive than any other species in the pot. And that makes a huge difference to what you can do compared to all the other methods, because you can have nucleophiles already present that can then open this episulfonium ion. And we can use azides to do that, or we can use a lot of other nucleophiles to do that. Here I'm showing you a few of them as weakly nucleophilic as anilines, and this is in water, all right? Once you go less nucleophilic than aniline, water will take over and you just get hydrolysis. Now you can say, well, I don't really care using an aniline to substitute a protein. But where it gets really interesting is that you don't have to use exogenous nucleophiles. You can also use nucleophiles that are already present in the environment of the proteins, such as another protein, right? And here I'm showing you one example where we identified a previously unappreciated protein-protein interaction. So there's two proteins together. The vinyl TT reacts with one cysteine, and then a nucleophile from a neighboring protein can just snapshot, and we link these two and can then analyze the structure of these protein-protein interactions and identify them. And the difference to other reagents that look at protein-protein interactions is that we only have an ethylene group, right? That's just a 1.3 angstrom bond that we have. Normally, you have long linkers, right? If you have something that reacts with a lysine and with another lysine, it reacts once, and then it swims through the cell, and then reacts with another one. And you have a very long linker. We don't. We have a very, very short linker, which makes this a rather powerful reagent because we can have very, very close interactions and say these things have been close already in the protein-protein interaction before. And that becomes even more exciting when we do this in cells, because we've discovered that this vinyl thiotrinium compound can enter cells. It goes through the cell membrane. It does that very efficiently, more efficiently than most other small molecules that we have compared them to, such as malamides or succinimides. And then it can do this chemistry in the cell and can look at protein-protein interactions in cells. Now, I am not a chemical biologist, and I most likely will never become one. But what is exciting to me in this area is that we can look at that reactivity, those potentially interesting protein-protein interactions in viable cells based on new chemistry that previously was not accessible. And that is something that, I really learned from Erick, that you can use new reaction chemistry that enables reactivity and applications that previously was just not possible. And that is something that I'm very excited about in this project right now, and we'll see where it gets us. And because I'm out of time, I want to leave it at that and have the most important slide here to thank all the students and postdocs who have done all the work that I showed you today and are working hard in the lab as we speak. Thank you again for Erick for making this event possible, and for Mark for hosting it. So I'll be happy to entertain any questions if there are any. Thank you.
Thank you. Thank you very much, Tobias. That was terrific. Really a wonderful lecture. There's a few questions that have come up. Some folks are saying congratulations and so on. So one question, is it possible to use the aryl thiotherium coupling chemistry for surface or material modification as compared to traditional methods that use diazonium salts?
Yeah, that's a wonderful question, because we have been working with our next speaker to do exactly that. Okay. So
that's a question from Hee-Jun Lee. So congratulations.
Not who asked that question, but yes,
that wasn't me.
Yes, we have quite an exciting collaboration, and the results hopefully will be forthcoming soon. That works.
Excellent.
And
I actually
don't want to say too much because it's too early. It works. It works very well.
I had a related question of my own, actually, which is just, can you stabilize the proteins more by cross-linking them in some way through this method? In other words, can you make them more thermally stable and so on?
So we have not, technically, absolutely, that should be possible. We have not shown that, but we have shown that with peptides. So if you take oligopeptides, and I think we've gone to about 20 mers, and I've done cross-linking of those, and yes, they have higher melting temperatures.
Great. And now another question from Dr. Soichiro Nakatsuka. Says the reaction mechanism on slide 10, the resting state, is the dissociation of tritibutylphosphine from the palladium bis tritibutylphosphine. Was there any reason not to use a 1-to-1 ratio? And then they just mentioned in Greg Fu's paper on developing using palladium tritibutylphosphine and KF, an experiment was conducted comparing palladium to tritibutylphosphine ratios of 1-to-2 and 1-to-1. It was found there should be no activation barrier at 1-to-1.
Yeah, that's true. So Greg showed that in a wonderful way. You can also just protonate one phosphine off. In that paper, I think it's HBF4 doing that. You can do that. In our reaction, that's not necessarily, because while it is the resting state, the turnover limiting step is actually the deprotonation of the dihydrogen. So you do not accelerate the rate, and the half order in the rate law only comes from the fact that one phosphine needs to dissociate, but you're not accelerating the reaction if you facilitate that step. So technically, mechanistically, absolutely yes. Practically, there's no benefit because the deprotonation of the dihydrogen on the palladium-2 dihydrogen complex is rate limiting in this case.
And one final question, maybe, can we control the position of thianthrenium salt formation from arene derivatives? What factors are there to consider and look into?
Yeah, that's a hard one to answer in a minute, because we published a 12-page JAX paper to answer exactly that question. The Hammett rho value of the thiantrination reaction is -11, which means that it's very, very sensitive to the electronic structure. So what happens is the rate-limiting deprotonation governs the regioselectivity. And so what it does, it goes to the most electron-rich CH bond. And that's also not something that we can change. That's inherent to the mechanism. So it will go to the most electron-rich CH bond. We can predict that. We always get it right predicting it, but we cannot say what we'd rather have it here. That doesn't work.
You have to take what you've got.
Yes, it's very selective, almost always, but that selectivity is inherent to the reaction mechanism, and we have not been able to change that so far.
Perfect. All right, I think we should stop there and thank Tobias very much for a very stimulating lecture, wonderful chemistry, and thank you for speaking on behalf of Erick today. We have like 350 people online for this talk, so that's really an excellent turnout. So thank you very much, Tobias.
Thank you.
Our next speaker is Professor Song Lin, who you can see on the screen. Song grew up in Tianjin, China. He earned his BSc from Peking University in 2008, and then he pursued graduate studies at Harvard. So there's the Harvard connection again, in the labs of Erick Jacobson. His doctoral research was focused on development and mechanistic understanding of enantioselective reactions catalyzed by multifunctional hydrogen bond donors. Motivated by deep interest in catalysis, he then did postdoctoral work with Chris Chang at UC Berkeley, and that research was focused on the design of molecularly tunable materials. So hence the materials is coming in again, including covalent organic frameworks and catalysts for electrochemical conversion of CO2 to value-added products. In the summer of 2016, Song moved to Ithaca, Cornell University, to start his independent career. He was promoted to associate professor in 2021 and full professor in 2023. So a warm welcome to Song, who's going to talk about enantioselective electrosynthesis. So please take it away.
Thanks, Mark. Can you see the slides? Okay.
I can. Perfect.
Great. Awesome. Thanks, Mark, for the kind introduction. And thanks for organizing this symposium here. It's a great pleasure for me to join the symposium to honor Erick's many great contributions to Thieme chemistry, but also to, as Mark mentioned earlier, to our community of organic chemistry in general. So I'm showing this picture here. So this is Erick given the presentation, and he's using his hand gestures to show am assuming some sort of structure or maybe even stereochemistry here in the presentation he's given. And indeed, that's one of the many great contributions that Erick has made to our field in terms of on the research side in the fields of asymmetric catalysis and stereoselective synthesis. And so today, I want to also talk about some of our contributions, particularly using electrochemistry to achieve enantioselective synthesis. So I guess before that, for those of you who don't know what we do, I just want to give a brief introduction to our research program. So we are an organic chemistry group, but we leverage electrochemistry as a sort of unconventional tool to study organic systems. So currently we have three main directions going on in the lab. The first direction is electrosynthesis, electrocatalysis. This is where we use electrochemistry as a tool to discover new reaction transformations that can be challenging to achieve using traditional means of activation. A second direction is electroanalysis. So this is where we use electrochemistry to understand the mechanism. So for example, using voltammetry and other electrochemical tools to understand the mechanism of reactions going through redox electron transfer pathways. And then we also have a third direction here, which is we call technology development. This is to develop reactors, particularly high throughput reactors, to facilitate the adoption of electrochemistry by the broader organic chemistry community. Now today, I'll primarily just focus on a couple of stories under the realm of electrosynthesis, so using electrochemistry to promote organic reactions. So I want to start by showing a story in the realm of alkene functionalization, asymmetric alkene functionalization, particularly alkene hydrogenation. I want to mention this work we carried out was actually really inspired by an early contribution from Erick's lab, who's contributed a series of very interesting papers and very important contributions in this realm of hydrogen atom transfer promoted hydrofunctionalization reactions, which now has exploded and become a very important sub-area of organic chemistry. Now, particularly, I'm just showing this one publication from him, I guess almost 20 years ago now, of this paper in Angewandte reporting the hydrogenation. So it's a cobalt catalyzed hydrogenation of alkenes, where they're able to use a hydrosilane and this tolzol cyanide as the reagents to add HCN across, essentially effectively HCN across the double bond. And so when we started to think about how to achieve electrochemical synthesis enantioselectively, we want to take a close look at this transformation here and see how we can induce enantioselectivity in this reaction. And so the idea we had after some exploration initially is to use a dual catalog system here. So this is the electrocatalytic system here where you have the electrode. This is the anode where the oxidation reaction happens. And so we're hoping to combine two different catalog cycles. One is a cobalt catalog cycle, which is inspired by Erick's work, and the other one's a copper cycle here. So in this case, the cobalt catalyst here, which, in this case, Erick used a cobalt saline. So this would undergo a single electron oxidation on the anode to give you a cobalt-3 species. And this species would undergo a reaction. You can, I guess, term this as transmetallation. And then this will undergo sort of essentially almost like a ligand swapping process where you can access this cobalt-3 hydride species. And so this intermediate is acidic. So the pK is around 10 or so. So this can undergo reaction with relatively nucleophilic alkenes to transfer the hydrogen atom to give you this carbon-centered radical species here. And so now we're hoping that this intermediate can then be passed on to a second catalog cycle here, where this copper can take over. So this copper-1 cyanide species undergo oxidation on the electrosurface to give you this copper-2 cyanide, which can undergo a single electron oxidative addition to give you a, I'm not showing the intermediates here, but presumably people propose a copper-3 alkyl species, which can undergo reductive elimination to give you this desired carbon bond. And so thinking about how to make this reaction enantioselective, we're hoping that if we can introduce a chiral ligand for copper for the second radical cyanation step, then we'll be able to synthesize these nitriles in high selectivity. And the reason why I want to achieve this particular reaction asymmetrically is because it turns out these nitriles, especially these benzylic benzylic nitriles are very well represented in pharmaceuticals. and their synthetic intermediates. So some of these nitriles, as well as their corresponding carboxylic acids or reduced amines, have been observed as important motifs in pharmaceuticals and synthetic intermediates. And many of these molecules turns out to be chiral. So to make the long story short, eventually we're able to get this reaction to work. So this was done by a very talented former postdoc, Lu Song, and she's now a professor back in China. So she was able to discover this catalytic system here, again mentioned earlier, using a dual cobalt and copper as the catalysts. So cobalt, we can just simply use a cobalt saline catalyst as the HAT catalyst, and then the cobalt for copper, we're able to develop a new ligand. So this box ligand with 2 ester group substituents, and with this new ligand under a relatively straightforward electrosynthesis conditions, we can achieve this hydrogenation reaction in very good selectivities. I'll talk about this particular ligand in just a second, but turns out this new ligand with the ester group with the serine-derived ligand is special in this case that gives very high selectivity. It turns out some of these classical box ligands that we can buy are easily synthesized, only give relatively low or moderate enantiomer selectivities. So just to briefly show you how the scope of the reaction, turns out a number of different conjugated alkenes can be applied to this reaction system. So most of the slides, the substrates I'm showing here are styrene type substrates. So for both terminal styrenes in these cases, as well as internal styrenes, can undergo hydrogenation reactions in good efficiency and selectivities. And we're excited to see that we can also use these vinyl boronase or vinyl acetase to give you these effectively difunctionized products in good selectivities. One thing I do want to mention is, I'll talk about this in a second as well, is this reaction, because we're using electrochemistry as a way to provide the oxidative equivalent, we don't have to use the typical traditional strong oxidants that have to be used to power such type of hydrofunctionalization reactions. So in this case, we can actually tolerate some pretty good functional groups. Some of them can potentially be prone to oxidation with the traditional strong oxidants, such as this electron-rich arenes, as well as thioethers and aldehydes. Now, in addition to styrenes, we can also apply this reaction to dienes and enines. So I'm showing you just several examples here. If you can take these conjugated dienes and in one case an enine here, we can also achieve the hydrogenation of the terminal alkene, the more sterically available alkene in generally good enantiomer selectivities. In a couple of cases we showed, for example, in this case, if we take an alene, we can depending on how many equivalents of your cyanide reagent you add, you can either stop at this mono hydrogenation stage to give you this allylic cyanide, or alternatively, you can do a second hydrogenation of the internal alkene to give you this 1,3-dinitrile in about over 90% E. Finally, we also did a radical probe experiment. So using the cyclopropane substitute alkene, you can then do the hydrogen atom transfer, and this will pop the radical will pop the ring open. And then benzyl radical would then be trapped by the copper cyanide intermediates to give you this species here in good selectivities. So after developing this reaction, we also want to understand how this reaction works, particularly how this new serine-derived box ligand gives us high selectivity in the reaction. And so this is when we teamed up with my colleague Rob DeStasio, who's a theorist at Cornell, and we did some computations. Actually, before that, I do want to mention earlier, before the mechanism, I want to talk about a couple of interesting features, again, tying up to the mechanism in a second, why we use electrochemistry in this reaction. Okay, so a couple of things we notice is, first of all, with electrochemistry, we know we can precisely control either the current or the potential you apply to the reaction system. That allows you to run these reactions to generate the reactive, for example, radical intermediates in a much more controlled fashion, in a selective fashion. And so one thing we've noticed in this reaction here is, I mentioned earlier, if you have electron-rich arenes such as this anisole or this indole derivative, what happens here is if you do the reaction using chemical oxidants, these can undergo degradation. And turns out even with electrochemical reactions, if you're not careful with the voltage you apply, so if you use a relatively high cell voltage, so this whole cell voltage about 2.3 volts, which is, I think, is about 500 to 600 millivolts on the anode surface, you can already start to see some degradation here. So in these cases, you see with higher voltage, you see lower yield and also formation of these side products where you potentially oxidize the carbon radical you're forming here to eventually to these ketone byproducts. And this can be potentially solved by just lowering the potential input. And indeed, if we apply a lower cell voltage here, about 1.8 volts, we can then increase the yield. In the second case, substantially increase the yield of the reaction and suppress the formation of these ketone overoxidation byproducts. The second thing is we want to test out whether, we use our expertise in electrochemistry. We use electrochemistry to discover this reaction, but we also want to see whether this reaction can be promoted by a traditional chemical oxidants. And we show that in this case, at least for this particular reaction here, a large number of chemical oxidants we try cannot be used to achieve this reaction with the same level of efficiency or selectivity. Okay. So we tried a number of different, single electron or 2-electron oxidants, as well as atom transfer catalysts. Oxidants. And in all of these cases, we show in many cases, we do show pretty good conversion of the starting material, but not necessarily to the desired hydrocyanation products. So you can see the yields are typically pretty low. In many cases, we do observe this benzophenone or some other similar overoxidation products. In the cases where we see some yield, we also see lower in-situ selectivity for reasons we don't fully understand just yet. But it's interesting to see how, at least for this hydrocyanation reaction, this is very unique to electrochemistry, and it cannot simply be promoted by a traditional chemical oxidant. And going back to the mechanism I mentioned earlier, this is a collaboration with Rob DeSasio, so his lab was able to compute the transition states for this particular reaction here, and particularly for this hydrocyanation, for the cyanation step, where you achieve the in-situ selective carbon-carbon bond formation reaction. In this case, I won't go into the details, but turns out a really interesting finding that his student Brian showed to us is that this reaction is controlled not only by this coordination with the carbon center, the carbon center radical, but also by interesting non-covalent interaction here. So we show that when you introduce ester groups here, so on the ligands, when you have these ester substituents here, you can make the alpha proton here much more acidic. So now this proton here can engage in the attractive CH pi interaction with the arene, with the pi system on our alkene substrate here. And we show that this hydrogen bonding, this CH pi interaction is very important to helping organizing the reaction transition states to achieve high in-situ selectivities. So that wraps up my first story, and now I want to move on to my second story. So I want to show you the reaction again. So this is the hydrocyanation reaction and some of the substrates that we showed earlier. In this case, we use this traditional way of doing interselective electrochemistry, which is to use a chiral catalyst. In this case, one chiral and one achiral catalyst. And this is indeed the most common way of achieving in-situ selectivity in electrosynthesis, okay, which is to use a redox active catalyst that is chiral, okay. And so you do what people call asymmetric electrocatalysis here. And so this is not surprising, where the catalyst can not only serve as electron shuttle, but also as a source for your chiral informations to make sure that the oxidation or reduction events can happen in an asymmetric fashion. So this is an interesting approach, and that has been proven fairly effective in the past several years. But this is not the only type of, this media type of electrosynthesis is not the only type of electrochemical reactions that we do. Turns out there's a very large class of reactions that people do using electrochemistry that do not require or do not need catalysts. Okay? And this is called direct electrolysis. Okay, so in this case, instead of using a mediator or catalyst, the oxidation reduction events happen directly on the electrosurface. So the substrate will just take up or give up electron on the electrosurface to become intermediates, which can then be transformed into products. Okay? So it turns out some of the classical organic electrochemical reactions, such as the Kobe electrolysis, where you do decarboxylative CC coupling reactions, or Schoenel oxidation, where you do CH oxidation reactions, both of these reactions are achieved using direct electrolysis. There are a number of really interesting recent examples from many labs around the world. But I just want to show you a couple examples here. Some recent contributions from, for example, from Phil Barron's lab. They call this an E-Birch reaction, so electrochemical arene reduction reaction. Also a contribution we made a couple of years ago using electrochemistry to achieve the cross-coupling of 2 alkyl halides. Okay? And both of these cases are also using direct electrolysis. Okay? So these reactions don't use catalysts, and you can argue sometimes you probably don't want a catalyst for various different reasons. So the question becomes, if we want to render some of these reactions asymmetric, how do we do this reaction? So this is a bit more challenging because now we can't or we don't use a catalyst. So we want to think about ways how we can, strategies how we can induce selectivity. And so we took a look at some of the components in the electrochemical system. So one thing I do want to mention is how electrochemical reactions are a bit different from traditional homogeneous reactions. It's heterogeneous in nature, and most of the reactions, especially when you generate highly reactive intermediates with very short lifetimes, the reactions mostly all happen on the electric surface at the interface. Okay, within this, some people call it a double layer. Okay, on the electrolyte interface. And so I want to take a look and see what this interface is composed of, right? So we can see some of the components of the interface and see how we can maybe use those components to induce asymmetry. So the first component is the electrode itself. Okay, so we thought about using maybe rendering the electrode asymmetric or chiral. And it turns out this has been done in a couple, I think in particularly one good examples. Which is something we're still very interested in doing. Someone mentioned earlier how we're working with Tobias. Tobias mentioned earlier we're working together to see whether we can use this as an effective approach to do chemical reactions, for example. But at least today, as an organic chemist, it's still not easy to think about how to make this process robust and also reproducible. Okay. So that's something we're keeping in mind, but not the approach we take for this particular instance. The second thing is the chiral, right? The solvent, right? The solvent is always going to be on the interface anywhere in the reaction. But we can use a chiral solvent, but that would be very costly and wouldn't be practical in today's organic synthesis. But then there's also a third component, which is electrolyte. Okay, so supporting electrolyte is a salt, typically either inorganic or organic salt we add to the electrolysis experiments to make sure the medium is conducting. And so in most cases, people think about chiral electrolyte as an innocuous salt that you add there that doesn't necessarily do anything to the chemistry you want to do in the reaction. And that is not always the case, even though people believe so, but turns out people have shown in the literature sometimes electrolyte can be highly influential in the reaction, in the reactivity. In this case, we want to see whether we can introduce a chiral version of the electrolyte, chiral salt, to render asymmetric render electrolysis reactions asymmetric. So the reaction particularly we want to achieve by using this approach is the dynamic kinetic resolution of trivalent phosphines. In this case, we want to take a racemic chirophosphine and be able to resolve it, ideally in a DKR, so convert both the enantiomers into a single enantiomeric phosphine oxide. And so in the interest of time, I won't give an extensive background introduction, but turns out this is important because first of all, these phosphines and chirophosphines and chirophosphine oxides have been used as ligands or organocatalysts in asymmetric synthesis themselves. But also recently, people have discovered some of these really interesting, chiral P5 complexes can be used in medicinal chemistry discoveries as well. So the approach we want to take is to do this DKR. And turns out this is relatively rarely explored in the literature. There's one really interesting example from Gil Heaney's lab from Ireland, and he was able to leverage this classical power reaction to do the DKR of trivalent phosphines. So in this case, you can treat this species with this this polychlorinated species here. In this case, they use triphosphine. Or actually, in this case, they use the hexachloroacetone, and you can chlorinate this phosphine here to give you this chlorophosphonium ion, which can then undergo rapid racemization through an additional elimination pathway in the presence of additional chloride. And this will then serve as the basis for the DKR, so you can resolve these two enantiomeric chlorophosphonium ions by adding a stoichiometric amount of this chiral alcohol. Eventually, you can get to this phosphine oxide in most cases in moderate to high enantiomer selectivities. So we want to further improve the enantiomer selectivity, but also develop a different approach to achieve this DKR oxidation process. And the idea we had is also based on literature. Okay, so we've all learned potentially from your sophomore organic chemistry that unlike amines, phosphines don't undergo this pyramidal inversion very easily. Okay, so this inversion barrier is about 30 to 40 kcals. But interestingly, it's been shown in the literature, if you oxidize the phosphine into a phosphine radical cation here, these radical cations, depending on what substituent you have, can undergo very rapid pyramidal inversion with a barrier of as low as 5 kcals. And so we want to use this rapid racemization of the oxidized radical cation here, potentially generate electrochemically to achieve the DKR process, where hopefully if the addition, the nucleophilic addition step to form the PO bond is fast for only one of the two enantiomers, then we can funnel all the material through this top pathway here to give you a single enantiomeric product, phosphate oxide product. Now, thinking about how to achieve this, the resolution process, because these radical cations are very reactive, the barrier for this nucleophilic addition is very, very low. It's hard to think about how to introduce a new catalyst to even further lower the barrier to catalyze this process. But with that, what we can do here is to introduce a chiral anion. So this cation is going to be associated with the anion, and the chiral anion can simply be added as part of electrolyte. Okay? And because we know these radical cations are generated on electric surface and they have such a short lifetime that they cannot escape the double layer before it reacts with the nucleophile, everything happens in this double layer here. And so we're hoping to introduce a chiral electrolyte to be able to induce the second step, nucleophilic addition steps, in a selective fashion. So one thing that works in our advantage about this approach is that it turns out because electrostatic interactions, the electrolyte is actually highly concentrated with this within this double layer. Okay, so for example, this reaction would be oxidation reaction here. So if you apply a oxidation current or a potential, the positive charge, this is naturally going to attract negative anions. Okay, so anions because of electrostatic interactions. And turns out there's some computational work by people showing that how within this double layer, the concentration of the electrolyte can be sometimes several times or even hundreds of times higher than in the bulk solution here. And this is great because this works in our advantage. We can pre-concentrate with this charged electrostatic interaction the current anion within this double layer, which can induce even a more chiral environment to achieve this selective transformation and combat the competing background racemic reaction. So again, to keep the story short, eventually postdoc Chen Fei and graduate student Kaining were able to get the reaction to work. So just by using the tetrabutyl ammonium salt of this commercially available chiral phosphoric acid, we're able to get this reaction to work. So using about 40 mole% of this chiral electrolyte, we can achieve the DKR of this trivalent phosphine to form these phosphine oxides in good selectivities. In this case, we use this nucleophile here, this benzamide as the nucleophile. In this case, this oxygen would attack the radical cation. Eventually, this deoxygenates and to become benzonitrile as the side product, which we can detect from the reaction. So you can see for this particular substrate here with this a hydrogen bonding donation group here, which can hydrogen bond potentially with your phosphate, you can achieve the reaction in good yield and very high initial selectivity. Here are just some other substrates that work in the reactions. You can change the substituents here on the phosphine or on the alkyl group on the directin group, the non-covalent directin groups here, and get the reaction to work in generally good selectivities. We also showed that this hydrogen bonding directin group is not necessary in the reaction. So we can also get the reaction to work for these relatively simple diaryl alkyl phosphines. In this case, we use this benzyl alcohol as the oxygen nucleophile, which undergoes a PO bond formation reaction. And in these cases, we get the reaction again to work in good selectivities. So in the interest of time, I'll just skip one slide and I want to just talk about the mechanism here very briefly. So in case you are curious about how this reaction works, so I mentioned earlier how you can oxidize the phosphine on the anode surface to this radical cation here. This will undergo rapid inversion, and then one of the enantiomers will then be oxidized to undergo nucleophilic attack in the presence of chiral electrolyte. So in the case of benzamide as the nucleophile, those undergo addition to the phosphors. You can undergo a second oxidation to give you this phosphonium, the oxyphosphonium ion intermediate, and this will then undergo essentially a polar beta elimination process to form benzonitrile and give you this phosphine oxide product. In the second pathway, if you use benzyl alcohol as a nucleophile, this will undergo addition as well, and then very rapid second oxidation here to give you a very similar alkoxyphosphonium intermediate. In this case, this phosphonium intermediate is relatively stable. So what we think is going on is this actually diffuses onto the cathode itself and gets reduced. So this benzylic CO bond will get reduced to give you the desired product. At the same time, you also form this toluene derivative, which we can detect as the main byproduct from the reaction system. We also did further DFT calculations to try and understand how the initial selectivities are induced. So we're very interested in seeing how this chiral electrolyte, not surprisingly, this chiral anion can not only be just providing the chiral environment, but also form hydrogen bonding interactions with a variety of different the nucleophile as well as the phosphoryl radical cation to induce a highly sort of organized transition state to achieve the DKR reaction effectively. I think that's at the time, so I'm gonna close the presentation by thanking the people who did the work. So the first work for hydrogenation reaction, this was done in collaboration with Rob Desasiu's lab and also with Mike Friedrich at Italy. So in my lab is Lou, Niankai, and Will who did the work. The second chiral electrolyte project was done by Kaining and Chenfei predominantly, but also with the assistance from Yi, Zhang, and Izzy, as well as Louise. This is also a very great collaboration through the Center for Synthetic Organic Electrochemistry with UHG's lab at Brown. Our lab, as well as the funding sources. And Mark, if we have some time, I'll be happy to answer any questions people might have.
Perfect. Thank you so much, Son. Terrific lecture. We appreciated it very much. So there are a number of questions. Let me just work my way through them. So one of them is, it says in the hydrocyanation, if lower potentials are beneficial to prevent side reactions, is it possible to run the reaction with constant current electrolysis? That's from Marcel.
Yes, so we have tried the reaction with both constant current and also constant potential. So we're in this case, we use constant cell voltage. So that's, you control the voltage between the two electrodes. But electrochemistry, you can also precisely control what potential you apply on one of the two electrodes, on the anode, for example. And so we tried that, and that also works. So if you just can do control potential electrolysis with a reference electrode, you can control precisely the anode potential. When you lower the potential, you can start to induce selectivity, obviously at the expense of lower reaction rate, but you can only challenges in that case, you have to use a reference electrode. So it's a bit less practical for large scale synthesis. So we decided to eventually use the control cell voltage approach for this reaction. We also tried control current. That approach is a bit less effective, but it also works.
Just on the hydrocyanation, there's another question about there's been a big push towards using less toxic cyanide reagents. Have you tried to get this reaction to work with other reagents, including, say, potassium ferrocyanide?
That's a very interesting question.
Wilson.
I think we tried potassium and sodium cyanide, which are, I guess they're solids, or they're a little bit easier to handle, and they do work to a certain extent, but not the yields are typically low, partly because of solubility. And we have not tried ferrocyanide. That's a really interesting question, something we should potentially try. I think, you can potentially generate cyanide source in situ that way by adding acid or maybe some sort of Lewis acid.
Okay, another question from Erick Alonso. Having to activate by oxidation both metal catalysts to promote the reaction, I assume it requires control of the redox potential via the ligands, meaning they should not be too far. Is that right? How much of a potential difference can one go and have the reaction succeed?
Right. Yeah. If I understand the question correctly, so you talk about the cobalt copper system, right? So we have two catalysts. And turns out in that case, the initial hypothesis, I didn't talk about the detailed mechanism, but initial hypothesis is you're oxidizing both catalysts on the anode. But turns out we did some CV experiments. We haven't published this yet, but it seems possible that one catalyst actually mediates the oxidation of the other catalyst. So it's possible that only one catalyst is oxidized on anode, but the other one gets the electron, or passes an electron from the other catalyst.
And Sarah's actually asked a question. So given the association of the phosphate with the electrode surface, could you imagine the electrochemical cell volume in terms of concentration of anion or size of the electrode could influence the selectivity? And has your team ever looked at this?
Right. That's something we want to look into, is use molecular dynamics simulation to, understand how, for example, the concentration and different electrode materials can influence the concentration of the electrolyte on the electrode surface. We haven't thought about it, actually. Interesting. So yeah, we can potentially use porous electrodes and see how that can maybe help further concentrate more of the electrolyte on the electrode surface. And we do have some data showing that, and this perhaps is not surprising, if you start to lower the electrolyte concentration, you start to see lower initial selectivity. But the drop is not drastic, and we're able to restore the initial selectivity by using a flow system or you have less volume to electron surface area ratio, and your electrodes are much closer to each other. In that case, we can use about 20 mol% catalyst while maintaining still pretty high selectivity. So there's definitely a lot of interesting aspects to explore there in terms of the electrode volume to electrode surface area ratio, the concentration of electrolyte in solution, even the electrode distance. So yeah, thanks. Interesting question.
Yeah. In fact, somebody followed up the question just saying that in a typical electrolytes are around 0.1 molar. It's surprising that such a low loading is able to work well. Do you have any comment about that?
I think part of it,
yeah. Sorry, go ahead, please.
Yeah, I think part of that is because of this concentration effect, where on the electrode at the interface, you have such a higher concentration, much higher than in the bulk solution. In our case, it's actually not, based on the MD simulation, it's not super high. It's like 3 or 4 times as high as in the bulk solution, but they can already allow you to the enantioselective pathway to be 3 or 4 times faster than the racemate background reaction. And there's definitely a lot we still don't understand yet. But I also do want to point out you're right. So the typical concentration of electrolyte is about 0.1 molar, but that really depends on the reaction, the medium as well. So if you use a relatively polar solvent and relatively polar electrolyte that dissociates really well, the concentration doesn't have to be as high.
I see. And someone else along that same line, have you looked at temperature effects on your chiral electrolyte-induced enantioselective reaction? They suggest it might be very strongly sensitive to that.
We did try that. We haven't tried increasing temperature. We tried lowering the temperature to see if we can bump up the selectivity a little bit more. But turns out it's actually not very effective. So even if you lower the temperature, the EE stays more or less the same, which is very interesting. Again, something we don't understand yet. It's possible that I guess a couple of things, right? One is that, for this, the temperature not only changes the chemical status, but also changes the interface structure as well. Okay, and that could have an effect on selectivity. But also, what I was going to say, the reaction. Yeah, I guess the temperature itself, yeah, maybe that's the main point. It might change not just the chemical process, but also electrochemical process. Something we want to try and I was going to say that the electrochemical process also is something we want to understand now, is that when you pass a current, you start to heat up the solution near the electrode as well. Okay? So people, at least in synthesis, don't necessarily know what's the real temperature on electrode surface. It's not super hot, by the fact that we can still see high selectivity, but the current will have this heating effect, and that's something that can complicate this temperature study too, and something we want to study. We have actually some ideas on how to study that.
And if you change the stirring speed, is there any effect there as well?
Not really. As long as it's fast enough. If it's too slow, maybe there's some effect, but yeah, we didn't see a huge effect on the stirring rate, which is good news, right? It means the reaction is at least relatively reproducible. You don't have to worry too much about the stirring rate.
Let's see if I can find one last question. What would be the estimated barrier of inversion for the phosphorus radical cation chiral anion pairs?
Yeah, we have not. That's interesting. That would be something interesting to study, right? We don't know. My guess is that it's definitely going to, just because of the computational data we have, that this does. You imagine if you don't have the transition state here, the hydrogen bond is also bound to the alcohol here. So if we take this out, just the phosphine radical cation and the chiral electrolyte here, I didn't mention this because of time, but what we uncover is this very interesting effect here where the nucleophilic oxygen here have hydrogen bond interaction with alpha proton next to the phosphorus, which is not surprising because once you have phosphorus radical cation, this alpha proton should be fairly acidic. Okay. And so not only is this just a Carreira anion, but also has some interaction with the phosphine, specific interaction with the phosphine radical cation. So that would potentially change the inversion barrier. We have not computed that yet. Imagine that could probably be energetically or computationally costly, but that's something we should probe. Very interesting question.
Very great. Okay, I think given the time constraints, we'll say a special thank you for terrific lecture, and also for the audience for lots of questions. So thank you very much, Song.
All right, thanks, Mark.
That takes us to our third and final lecture for wherever you are. For me, it's this morning, and especially for Sarah, it is really this morning. So I think her devotion to this talk is to be recognized, given that it was 6 a.m. in California time. I said British Columbia. That was the Canadian equivalent. So a little bit of news about Sarah. Sarah completed a Bachelor of Arts in Chemistry from Connecticut College, then did a PhD in chemistry from Yale University. Following postdoc work at Harvard, she joined the faculty of Caltech, where she's now the Bren Professor of Chemistry and the Norman Davidson Leadership Chair for the Division of Chemistry and Chemical Engineering. She served as an editorial board member for Organic Synthesis, currently an associate editor for Journal of the American Chemical Society. She's also the chair of the department, and as chair, I can say that's an accomplishment to be doing all she's doing and running Caltech. Reisman, that is Sarah, has been recognized with many, many awards, teaching and research, Alfred P. Sloan, Cottrell Scholar, Arthur C. Cope, and Tetrahedron Young Investigator, and the ACS E.J. Corey Award, along with the Mukayama Award from Japan. So next up is Sarah. Her lecture is Necessity is the Mother of Invention, Natural Products and the Chemistry They Inspire. So thank you very much for agreeing, Sarah, and take it away. We can't hear you, by the way. I don't know if you're on mute.
I am unmuting. Sorry, I had already gone into laser point mode, which slowed down my response rate there. So can everyone hear me now?
Yes.
Great.
Perfect.
All right. Well, thank you, Mark, for the very kind introduction. And I want to echo the comments from both Song and Tobias about just acknowledging Erick's service to our community in a general sense through his work with Thieme, but also the ACS. And at a personal level, I just want to make a comment, especially for the students on this call, that it's really important. You have your graduate advisor, your postdoctoral advisor, but it's important to find people who support you beyond that immediate network, right? So your advocates in the broader community. And for me, I've always felt like Erick is someone who's been extremely supportive of my program, especially in natural product synthesis. He asked me to serve at Organic Letters and then again at JAX, and I'm just indebted to his support for my program. And so anytime I get asked to do anything on behalf of Erick, I'll say yes, because I think that he has really been very generous with his time and his support for me and the broader community. So
Sarah, apologies, we don't see you.
You don't see me. Oh, no. Okay. I'll try one more time. You don't need to see me, but let me get out of my laser pointer. So, oops, everyone will be able to tell that it's early morning here by the tired eyes. Okay, so now hopefully all technical aspects sorted out. I'll tell you a little bit about our program. And as you've already heard about today, I think probably needs very little introduction here for this audience. Any time you have a small molecule drug, you're looking at compounds, that are either designed or often, historically have been inspired by or derived from natural products. And so even in an era where you see, an increasing number of these fully designed synthetic compounds, we still have very interesting molecules being brought to market that are based on natural product synthesis. For example, this compound eribulin, which is a derivative of halachondrin, and the synthesis was developed by Kishi at Harvard and then brought to market by Eisai. And there's a tremendous amount of work that goes into developing the synthetic methods, also all of the sort of technical aspects of working with complex molecules on scale. But I think that, you know, the recent the recent use of eribulin as a therapeutic on the market, right, really underscores the importance and the ongoing importance of both natural product synthesis and reaction development in an academic lab. So my group is interested in the synthesis of complex natural products. That's really often a starting point in our lab. We think about it from a target-based strategy development. So a lot of times we're asking, what are key strategic bonds that we could disconnect that allow for, say, modular assembly of these compounds? And then what are the tactics that are required to enable these types of strategies to be implemented? So we have a number of people who are working in natural product synthesis at the same time, and certainly in concert with these efforts, we have a program in reaction development. And so we've had a number of papers in the area of nickel catalyzed reductive cross-coupling with an emphasis on asymmetric catalysis. More recently, we've been working on the development of catalytic methods, catalytic reductive couplings using samarium. And then all of these efforts at this stage are sort of leading us to ask how we can implement emerging tools from machine learning to guide both our natural product synthesis efforts. How do we de-risk complex synthesis? And how do we use them to enhance the optimization of catalytic reactions, especially when you're thinking about asymmetric catalysis and having to go through this empirical process of chiral ligand design? How can we help use modeling to guide those types of efforts? Now, because this is a symposium that's honoring Erick's contributions, I thought I would focus on a natural product synthesis. I think one of the things that Erick has done really beautifully over the course of his career is show how synthetic methods development can enable natural product total synthesis. And so we certainly aspire to do the same thing, right? To think about either developing our own methods or often leveraging emerging methods from the community and asking how we can use them to address key strategic problems. So the family of natural names are diterpenes. And I show here just sort of a selection of diterpenes with this canonical biosynthetic pathway where you have geranyl diphosphate. And depending on your enzyme, your synthase enzyme, you can cyclize to give different scaffolds. I'm showing here N-carine and N-atosane. And then these can be elaborated through further oxidations and rearrangements to various natural products, right? And so there are people who are very much motivated by the biosynthesis of these diterpenes in terms of strategy design, asking how do we emulate, say, the oxidase phase of these types of biosynthetic pathways. Our group has been interested in a slightly different approach in that we want to ask, how do we sort of think about accessing these diterpenes through fragment coupling approaches? And so instead of doing the cyclization, you might imagine through the central bonds that I've highlighted here in each target, or the central rings, I should say, and then be able to have sort of a modular access to these types of compounds. So I'll show this again in this way where I've kind of stripped the functionality and I just show some of the frameworks here. And one of the things that you can see is that, for example, from the grainanes to the N-carines and also in many of the diterpenoid alkaloids, there are common motifs that show up in these scaffolds. And so if you could develop, say, a collection of building blocks that sort of cover a reasonable chemical space of diterpenes, and then you could identify what are the sort of most strategic tactics to assemble these building blocks, you might have a very programmable approach to accessing various diterpenes. And I think that if you can do this, then you can actually streamline the synthesis. You can be able to access different compounds. And maybe if you're interested in biological evaluation, it can work in the grainanes. Although I want to say that in a broader context, we're interested in many different diterpenes. And I also want to say that I think that although this project I'm talking about right now sort of precedes some of these ideas and the tactics that we're using, It's very much this idea that continues to motivate some of our ongoing work in cross-coupling chemistry, because I think cross-coupling can be very powerful as the CC bond forming steps to sort of join these ring systems. And you will see that a cross-coupling reaction does, or an intramolecular cross-coupling reaction plays a key role in the synthesis I'll talk about here. Okay, so as an example of how I think fragment coupling approaches can be leveraged to access complex diterpenes, I'm just going to show here some prior work from our lab on the synthesis of persianol. And so in this synthesis, we were able to access two building blocks here, both materials. We can perform a 1,2 addition of this organolithium into the aldehyde to join the two fragments. And then we perform a second step, this palladium catalyzed carbopalladation carbonylation cascade to form the lactone. You can see here that we have much of the core, we have the core of persianol. We require the formation of one more CC bond to access the target here. But in this sort of two-step sequence where we couple the fragments and then perform this transition metal catalyzed reaction, we assemble quite a bit of the complexity of this skeleton. And this, I really think it was this project that got us started, got us thinking about the various ways that we can both do sort of fragment couplings and then, what you might call a scaffold tailoring approach, or this intramolecular cyclization approach using cross-coupling or catalysis to form natural product cores. All right, so building blocks that we've made in our lab that we think can be used in a sort of modular way to assemble different variations here. And I'll talk about today our work on the grainane. So I will keep the introduction brief. There has actually been a lot of work, so I'll apologize that I won't go into a tremendous amount of detail for other contributions. But we've been interested in the grainane. So grain toxin was isolated in the last century, a long time ago, 1912. Structure was not fully elucidated till the 1950s. And these compounds are interesting. They're produced by the rhododendron, but they typically are inhibitors of voltage-gated sodium ion channels. So, this is interesting if you have an interest in non-opioid analgesic type activity. And so I think given the challenges with opioids, if you could access compounds like this or something that could make it to the market right now, but I think that there is an interest in these types of compounds for that reason. All right, so people have been working on the grainanes for a long time, and the first actual synthesis was reported in 1976. This was a relay synthesis. The first total synthesis was reported by Shirahama back in 1994. The Shirahama synthesis really focused on this key carbon-carbon bond through an intramolecular pinnacle-type coupling. There was actually, I think, as was the case with many of these diterpenes, there was some initial work, beautiful synthetic work, pretty high step count, but beautiful work. And then there was a period of dormancy, and it was really, in 2019 where we start to see this resurgence in synthetic efforts towards this family of natural products. So beautiful work from Tim Newhouse and Haifeng Ding on the compounds shown here. And in the intervening time, there have been a number of total syntheses that have been reported. Now, we've been targeting this compound arequitol A. It has an additional ring here in the form of this hemiacetal, or this acetal, cyclic acetal. We thought that we might leverage that extra ring as part of our strategy. And so I'll show you that in just a couple of slides. This was isolated in 2019, so it's a relatively recent addition to the family of grainanes. It's isolated in low yields, and there have been no completed total syntheses. That said, we recognize that we have to, be aware of the contributions for all these related natural products. And I'll also just point out, and this is important, that the stereochemistry here, the relative stereochemistry that you see is important. So you can see in grainotoxins 2 and 3 that you have this sort of anti-relationship. Whereas in principinol D, this is a synthesis that was reported from the Newhouse group. You have this syn relationship here. Okay, so this was a project that was started by a postdoc, Carter Farley, but it was really led largely, I would say, for the last several years by Jordan Thompson, who just finished up this summer in the lab and is starting a position at Bristol Myers Squibb next month. Jordan was a mentor to a younger graduate student, Kayla Youngblood, as well as a visiting student from the Carreira lab, actually, so Sean Thieme, and then an undergraduate student at Caltech, Gary Zhang. Okay, so let's go ahead and talk about our equilitol. I've highlighted here, in terms of the strategy, this central ring. I think some of the diterpenes that have more complicated bridging systems, maybe the initial disconnection is a little bit more challenging to conceptualize. I think for the guanines, it's a little bit more straightforward, right? Ideally, what we want to do is basically disconnect the molecule through this central 7-membered ring. Now, you still have a few options, but one of the things that you might learn if you read Corey's Logic of Total Synthesis is that it can be strategic to disconnect compounds where you actually cleave the ring that has the most highly bridging ring system. And I've shown here the sort of central 7-membered rings. I've highlighted the 2 bridging carbons here in blue. And our goal was really to disconnect through these bonds. So we want to disconnect through the 2 bonds that connect a blue and purple atom. We thought that that would allow us sort of the most modular synthesis. It would address some of the stereochemical questions directly. And the challenge with that is that you have to think about which step is going to be your fragment coupling step and what step you're going to actually, let's say, use transition metal catalysis to forge the bond. And so if we do a little further analysis, I'm showing here sort of these charge affinity patterns that have been popularized by Dave Evans. And what you see is that you have this 1,2 dissonant oxidation pattern here. And that's why in the original synthesis of guanetoxin, actually Shirahama forms that functional group array through a pinnacle coupling. That's an UMPL tactic. And it allows you to access a CC bond with both of your oxidation. Now, they use samarium diiodide. We love samarium diiodide. That's a great reagent, but these types of intermolecular reactions don't work very well. So that would mean that they do it at the very end of the synthesis in an intramolecular reaction. And we've used similar tactics, and that is effective. But that means that then you have to form this bond here through some sort of cross-coupling or some sort of CC bond forming step. We thought for our equilitol that we might actually be able to leverage this key CC bond formation as our fragment coupling by using this butenolide as a building block. And what we like about the butenolide is that you have the ability to deprotonate, and that means that this position, although it's next to an oxygen, and electrophilic, is also nucleophilic. And so we can leverage the amphiphilic reactivity perhaps to engage with an aldehyde building block such as this one here in our coupling step, our fragment coupling step. Now, what we also know is that for these butenolides, so for example, if you convert your butenolide into a siloxyfuran, as you'll see, there's a strong preference for formation here at this gamma position versus the alpha position, even when you have substitution next door. And we thought that would be important because we're going to try and form a CC bond next to this gem dimethyl group. So this is sort of the conceptual approach. We thought that we could use the butenolide. We're going to leverage nucleophilic reactivity here to join, form a bond between the butenolide and the aldehyde. And then you can see that I've incorporated an alkenyl halide at this position, which we thought we could use to form this second CC bond through a cross-coupling step. So if I take you to a more complicated or a more detailed, I should say, retrosynthesis, this is what it looks like. We're going to use enolate alkynylation to forge this bond. That takes us back to a intermediate like this. And we're going to use a vinylogous Mukayama aldol type reaction to form this bond that I've highlighted here in blue. So that requires then the synthesis of both of these building blocks in an androgen-rich form. Now, while we were thinking about how to make this particular sort of complex aldehyde, we recognized that we could do some proof of concept chemistry with this aryl halide. This is available in 2 steps from commercially available materials, and that would allow us to sort of develop the vinylogous mukayama aldol, as well as do some initial scouting reactions for the enolate. Instead of alkylation, this would be an arylation. So here's the synthesis of our butenolide. We leverage Noyori type hydrogenation to access this enantiomer-enriched alcohol. This is a beautiful reaction. It was initially on this substrate disclosed by Olivier Baudouin. We then were able to protect the alcohol as a silyl ether. We alkylate with ethyl bromoacetate, hydrolyze this ester, and then we use thionyl chloride to form this chlorolactone. And what's neat about this intermediate is actually you have all the oxidation that we need for the final butenolide. What we can do is treat with base. You can eliminate the chloride and then isomerize the resulting olefin to give us this compound here in 6 steps from commercially available material. So with this in hand, we could start to look at the fragment coupling step. We did try some initial conditions directly from the butenolide, but found much more success by first converting it to this siloxydiene or the siloxyfuran. And ultimately, what we found is that zinc Lewis acids gave us the best reactivity. So the yield is still, somewhat modest, and I think that is really attributed to the fact that you have somewhat sluggish reactivity here next to this quaternary center, and you do have the possibility of competing sort of silyl ether formation at the aldehyde. Sometimes that's something that we can see grow in. But we did see very good selectivity in this reaction for CC bond formation adjacent to the quaternary center, so at this gamma position versus at the corresponding alpha position. We see good diastereocontrol control guided by the siloxy ether at this position, and we see good diastereocontrol control in this case at the alcohol, although it gives us the incorrect stereochemistry for direct transformation to orichalotol, but we'll invert that later in the synthesis. Okay, so we can now elaborate through a protection and reduction step to give us the substrate here, and so we can look at the enolate arylation. And we looked at a few different conditions. We didn't start with nickel, but ultimately we found good promising reactivity with some conditions first reported by Buchwald using this nickel catalyzed enolate arylation, and we could see formation of the 7-membered ring. Now, what was interesting is that under these reaction conditions, we were also seeing some of this undesired, what we call beta addition product. And this was, more prevalent with ligands like DAVPHOS, and we could bias towards our desired reaction by using bidentate phosphines like DPPP. So mechanistically, what we propose is that with phosphine ligands, you can have, in the aryl bromide, you can have oxidative addition of the aryl halide to give this nickel 2 species. Which can undergo transmetallation. And in our desired scenario, you're going to have reductive elimination directly to give you the CC bond. But if you don't have, let's say, a bidentate ligand, and you have the ability for an agostic interaction with a CH bond, you can get beta hydride elimination and then reinsertion to give the 6-membered ring. And so when you have phosphine ligands, you can kind of use your ligand to guide the reactivity towards the desired 7-membered ring formation. So this was a sufficient proof of concept for us to then move on to the real system. I'll go in the interest of time somewhat quickly here. We've developed a synthesis of the building block that leverages my colleague Brian Stoltz's enantioselective allylic alkylation chemistry. And that provides us with this alkenyl bromide here in reasonable EE. We can then deprotect the ketal and perform a nickel catalyzed 1,2 addition to give us this sort of 3-2-1 bridging bicycle. What's interesting about this is that by using tridentate terpy as a ligand, we can favor this 1,2 addition to form the 5-membered ring and outcompete Heck chemistry that happens with the alkene or 1,2 addition to form the 4-membered ring. We can then elaborate this through first protection, formation of the enol triflate, and then we use this alkene here as a precursor to our aldehyde. So we do an oxidative cleavage and then a halide exchange. We're going to use this alkenyl bromide. I can assure you that we looked at using the alkenyl triflate also directly, but that was not successful in the next nickel step. Okay, we can use the same chemistry here as before for this vinylogous mucayama aldol. I'm showing here 48% yield of this product. We do see, again, good diastereoselectivity selectivity at this position. I should have it on here, slightly eroded DR at the alcohol, although that's inconsequential. We can then perform a silation, and then the same copper hydride reduction gives us this lactone. Now, this is where we started to run into some challenges. We already knew that we had to worry about this alpha alkynylation versus beta addition. But with the alkynylation chemistry, this was a lot more challenging to sort of guide towards the desired 7-membered ring formation. We see a lot of this beta addition product, even with the phosphines that we had previously identified. Moreover, what we found is that these reactions were quite irreproducible. And like the harder we tried to make like well-defined nickel phosphine complexes, the worse the yield was. And it was around this time that Jordan was reading the literature and thinking about some recent work from Carreira, looking at these low temperature Kumada couplings, in which they propose a mechanism using the 16 electron nickel 0 complex that first involves formation of this 8-type complex and then oxidative addition followed by reductive elimination. And they invoke this as being very important in the reaction for this alkenyl bromide. And so that led us to ask the question about maybe when we were doing some of the initial chemistry, phosphine wasn't actually important for the alkenyl halide system, that maybe we have this mechanism shift where you need to access these sort of nickel 0 olefin type complexes, and those are what's reactive in the system. And so we started looking at various nickel olefin complexes, in particular this 16 electron complex reported from the Engel group performs much better in the reaction, favoring the selectivity for this alpha alkynylation. So now we're seeing a good ratio of the desired product to the beta elimination. And so although we have yet to do rigorous mechanistic studies, what we're thinking about is that as you go from the arylation to the alkynylation, you have this sort of shift in mechanism from an oxidative addition first to an 8-complex first type mechanism. We've done a little bit of optimization here of that reaction. So you can see this is what it looks like in the sequence to form the 7-membered ring. We're here in 13 steps in the longest linear sequence from the cyclopentanone. And we have much of what we require for the target. I realize that I'm running out of time. So I'll just show you sort of what we have to do. We need to perform a hydration here of this exoolefin. We need to reduce the double bond. We need to reduce the lactone to and form the ketone, and then we need to invert. And the way that we do this is first through a highly diastereoselective Mucayama type hydration of the olefin that gives us this tertiary alcohol. We then treat with acid to deprotect this TMS ether. We need to do this selectively in the presence of the TBS ether. And then we oxidize to the ketone and reduce both the alcohol, or the ketone and the lactone at the same time. So you can see that we've corrected the stereochemistry here. Very good DR for that step. And we can also form this lactol. So then we're able to complete the synthesis first through exchanging the lactol to the acetal, and then using an M-hat type reduction, and we're able to form the desired product here. It's formed as a 1-to-1 mixture with the 9-epi compound. I'll confess that we've tried this reduction on lots of other substrates, and this is the one that gives the best diastereoselectivity here at this bridgehead ring fusion. And so, we were very pleased to be able to access the natural product through this final sequence. And you can imagine that you do have the tertiary alcohol here that starts to block that top face. So, so this actually worked out well. And we've just recently finished a synthesis. We're getting ready to submit this work for publication. So I'll just close by returning to this slide here and give you a sense of how we're thinking about these targets and trying to think about the various types of fragment couplings and then transition metal catalyzed methods that we can use for cyclization that we think could access, actually from some of these common building blocks, multiple different families of natural products. And that's something that we're very interested in. I'll just close by thanking my group. I want to thank the NIH for funding this chemistry for funding total synthesis. People hear that that doesn't happen, but it can still happen in the US. Well, it could until recently, maybe, we'll see. I need to acknowledge many collaborators that we've been working with recently, although I did not talk about those projects today, and then my group, and I'm happy to take any questions.
Thank you very much, Sarah. That was terrific, wonderful discussion about strategy and wonderful information about new reactions. So a couple of questions in the chat. One is from Nicholas Wilson, just asking, in the nickel catalyzed reactions, have you tried making pre-made nickel oxidative addition products?
You know, not for the alkylation system. I mean, I think that's an interesting idea that we could look at. I mean, I will say that we usually those syntheses happen on larger scale, and it is, you know, at a stage in the synthesis where material has been a little bit precious. Although now that we have things worked out, we might be able to do that. But no, we have not at this time.
Okay, great, thank you. And somebody else, you mentioned that the triflate didn't work, and you'd certainly tried it. So you made it the bromide. Any thoughts or comments on why that was the case? What did you learn?
I think it was stability of the triflate under the conditions, and in particular in the presence of both the base and the Lewis acid there. We were seeing some formation, I think, of the ketone, so hydrolysis type products. But yeah, we think that that particular enol triflate is just not sufficiently robust under those reaction conditions.
Great, thank you very much. One other person was just noting that there's no public preprint yet. So I guess this is really so hot off the press, it's not in the press. Is that correct?
It's not in the press yet, which is why there may be an embargo on the recorded version of this talk showing up. But I was very excited, and I wanted to talk about this total synthesis because, you know, felt like for a Carreira honoring symposium, that would be fun. But the draft of the paper is in place, and we're just doing the last final wrap up. So it should show up on the preprint server soon.
Maybe we'll embargo my introduction as well. And one other question, is the structure determination mainly done using NMR, or somebody commented about machine learning for NMR data of diterpenes also seems interesting.
Yeah, that's a good point. And people are definitely leveraging computational tools for structure assignment. Most of this was by NMR. And once you get to these ring closed systems, you can get some really insightful NOE experiments. We do have crystal structures. In particular, we were able to get a crystal structure of the aerolation product, intramolecular aerolation early on, that allowed us to sort of confirm the stereochemistry at the secondary alcohol and adjacent to the quaternary center. And then I think we have some other crystal structures at various points along the way. It's always nice to get a crystal to kind of validate our NMR assignments.
Right. And one final question. This one's actually from one of my students. It seems that the bridgehead oxidation seems to be particularly challenging. Did you look at other strategies besides the nickel catalyzed version?
You know, we have looked at a few different ways to make that building block. In fact, our initial route involved what I would call a Snyder type cyclization and oxidative cyclization, which is very effective for 3-2-1 synthesis, but is much more sensitive to that bridgehead oxidation and is not enantioselective. And so we're doing this like preparative HPLC that was limiting material. So we ended up at the Stoltz alylation because we can do that on scale. We can actually process a lot more material, although the synthesis is maybe one step longer. We do, we have an alternative strategy right now where we can actually form that through a pinnacle type coupling using samarium. And so that looks pretty interesting. But then we have to, it probably isn't going to show up in the paper because we'd have to go back and retool our protecting group strategy a little bit. And so we have looked at other things, but I think that this is actually a really nice demonstration of how, Corey had done some of these intramolecular 1,2 additions into ketones using cuprates. The nickel chemistry is much more mild and much more, you can get good chemoselectivity over these other competing processes with the right ligands.
Great, thank you very much. Now, one thing some folks may not know, Erick was able to join. So he sent a note through the chat just saying he's been enjoying the lecture. What I don't know is whether Erick is able to unmute and say anything. So Erick, I'll just ask you if you want to. We're happy to hear from you. Oh, there you are. There he is, live and in person from China.
Not ideal. It's 11 p.m. over here, but it was great to hear the science. I'm grateful to the wonderful science that was presented today by Sarah, Song, and Tobias. They underscore the vitality of our field that evolves and reinvents itself, right? And it's great to be a part of that, with the focus being, of course, on new reactions and that lead to new strategies and new tactics. Thanks, Mark, for hosting that. And of course, thanks to Thieme also for making this possible. We should do more of these, I think. It's great to be able to share science across long distances and time zones. And so let's do more of these.
It's amazing to see how many people joined. We had about 450 sign up and 350 at various times attending, and we've got pretty much every part of the globe covered as far as we can determine. So thank you. And I think that's a real testament, Erick, to the reach that you've had, and it's a testament to the amazing people you suggested who would be speakers and who all agreed. So at all different time zones, they were in for the event. So thank you. Jeanne, do you want to say anything at the end as the Thieme representative?
Thanks, Mark. And thank you, Erick. Thank you for inspiring us and for inspiring all of the great science that was presented today as well. We heard so many fascinating findings from our speakers. I wanted to thank Tobias and Sarah and Song, and most of all, I want to thank Mark for chairing today. I want to thank everybody for making time and coming along. And I do agree with Erick, we should do this more often. And this is such a great way to share science across the globe and across different time zones. And there are no barriers to joining and sharing science. Thank you so much. This has been a great honor for us to host this special event, and we hope, Erick, that this has been a small way of us honoring you and thanking you today.
So I'll just say then goodbye to everyone. Erick, if you can stay on just for a second, please, you don't have to go, but just we'll say good thanks to all the speakers. Yeah. And thank you all for joining us. You can see all you need to do to get a symposium is put in 30 years at Thieme, publish a few hundred papers, train a few hundred students, and you too can have a Thieme symposium. So thanks everyone. Bye-bye, except for the
Sarah.
speakers.
Thanks, Song. Thanks, Tobias. Thanks, Jenny. Thanks, Jeff. I saw a number of people. Thanks, Sydney.